Question number 36. Function of lenticel is, the given options are A. Transpiration, B. Conduction of food materials, C. Gaseous exchange, and D. Both A and C. So here this is about lenticels. Lenticels are special structures which are being seen on outer surface of matured stems. So when a dicot stem will undergo secondary growth, it will produce bark on their outer surfaces. So the bark, below the bark, the cork tissue is present, which is impermeable to gases and water. So as a plant requires gases and water from outer surface, so it will produce some special lens shaped structures called as lenticels. And these lenticels are the lens shaped structures which are filled with loose mass of parenchymatous tissue. That loose mass of parenchymatous tissue is called as complementary cells. And as these cells are loosely arranged, and these cells are generally non-lignified in their nature, and as they are loosely arranged, they will allow the exchange of gases. That is, they are helping in gaseous exchange, the main function to be considered here. And apart from that one, it is also concerned with a little amount of transpiration also. Generally, out of the entire rate of transpiration performed by the plant, 1 to 2% of transpiration is done by lenticels in the matured plants. So these are the two functions which are being carried out by the lenticels. So from the given options, we can say that A and C are the correct options. So option D is the correct answer. Question number 37. In a woody dicotyledonous tree, which of the following parts will mainly consist of primary tissues? The given options are A. all parts, B. stem and root, C. flowers, fruits and leaves, and D. shoot tips and root tips. So here primary tissue. Here primary tissue may be seen in majority of the plant parts, but here mainly in which part of the plant body does this primary tissue are present. So these are mainly been seen in the tips of the plant parts, like they have been seen in the shoot tips, that is tips of the stem, and also in the tips of the branches. And apart from that one, they can also be seen in the tips of the root, that is in the root tip. So these two parts are completely been filled by the primary tissue. That's why we can say that mainly consists of primary tissue in the shoot tip and the root tip. So from the given options, option D is the correct answer. Question number 38. The function of a vessel is, the given options are A. to provide stability, B. conduction of water and minerals, C. conduction of food only, D. all of the above. So here vessel is the part of xylem tissue, and as we know that xylem is a tissue, or xylem is an dead conducting tissue. It's a dead tissue and concerned with the conduction, that is transportation of materials from one part of the plant body to the other part of the plant body. And here the conduction may either contain the food material conduction, water conduction, or mineral conduction. As the vessels are dead tissue, the protoplasm is absent, or they lack protoplasm. As the protoplasm is absent, they cannot conduct the food materials, and in place of the protoplasm, they have an empty space called as lumen. That empty space is concerned with the transportation of water and minerals from one part of the plant body to the other part of the plant body. or otherwise we can say that when the root hairs are absorbing the water from the soil, from there they are conducted through different parts of the root, that is epidermis, cortex, endodermis, and finally it will reach into the stele, that is into the xylem part, and from there the upward conduction, or we can say ascent of sap is taking place. That is ascent means upward movement, sap means here water along with the mineral nutrients is called as a sap. That is upward movement of the water and minerals is carried out by the main water conducting tissue, that is called as vessels. So from the given options we can say that option B is the correct answer. Question number 39. The youngest layer of secondary xylem in the wood of dicot plant is located between. The given options are A. Pith and primary xylem, B. Just outside vascular cambium, C. Just inside vascular cambium, and D. Just inside cork cambium. So here when you talk about the cambial ring or vascular cambium, we can call it as vascular cambial ring. So this cambium will start producing some tissues towards outside, that is called as secondary phloem, and some tissue towards inside, this is called as a secondary xylem, or secondary xylem is otherwise called as a wood tissue also. Whatever the secondary xylem that is formed, that will be gradually pushed towards inner side, such that the place is further occupied by the youngest xylem, which is being formed in the course of its growth and development. So what is the position of the youngest xylem that is formed, youngest secondary xylem or youngest wood that is formed during the secondary growth means it is present just below the vascular cambium. So just inside the vascular cambium. That is the reason why from the given options we can say that option C is the correct answer. Question number 40. The vascular bundles of monocot root are. The given options are A. Endarch radial, B. Exarch radial, C. Endarch scattered, and D. Exarch bicollateral. So here it is about a monocot root. So here if you observe the characterization, all roots will exhibit exarch condition of the xylem, that is arrangement of the xylem will be in the exarch condition. So here the exarch condition, and of course the stems will show the endarch condition. The differentiation is because of arrangement of the protoxylem tissue. Here in exarch condition, the protoxylem is towards outer side and the metaxylem is towards inner side. So this is a primary condition that can be seen in it can be seen in both the monocot root and also in the dicot roots. Now let us see the internal organization of the root in a ground plant. So this is epidermis and this is a stellar part. In the stellar part we can see that xylem and phloem are present on a separate radii, that is the xylem is in the separate bundle and phloem is in the separate bundle. If you observe, this is a protoxylem tissue and gradually the size of the xylem tissue will be increasing towards inner side. So this is a metaxylem and this is a protoxylem, and here also protoxylem, and the size of the xylem tissue will be increasing towards the center, that is metaxylem. So similar type of arrangement can be seen here with the tetrarch condition of the xylem tissue, and this part is occupied by the phloem. Like this, if we take, and this part is shown with the other xylem tissues also, like this, more than 6 to 7 xylem bundles should be there. This type of arrangement can be seen in the monocot root, and in between them we can see the phloem tissue here. So in general, in monocot root, we can see the polyarch condition, that is many xylem bundles are present. And here the xylem is in one radius and the phloem is on the other radius. So we can call it as separate or radial vascular bundles. That means here in the monocot root we can see exarch condition and radial condition. So from the given options, option B is the correct answer related to monocot root. Question number 41. Who differentiates leaf of dicots from monocots? The given options: A. Parallel venation, B. Differentiation of palisade and spongy parenchyma, C. Stomata only on upper side, D. Stomata both on upper and lower sides. So here the differentiation of the dicot leaf from that of the monocot leaf. That means here the character should be exhibited by the dicot leaf, but it should not be present in the monocot leaf. So let us take the first character, that is A. Parallel venation. Here parallel venation is a characteristic feature of monocotyledonous plants. So this is not present in dicots but present in monocots. So as we know that dicots exhibit reticulate type of venation. And B. Differentiation of palisade and spongy parenchyma, that is the ground tissue of the leaf, that is mesophyll, is differentiated into two types of tissues, that is palisade parenchyma and the spongy parenchyma. So this type of nature can be seen in dicot leaf, but in monocotyledonous leaf either they have spongy parenchyma or palisade parenchyma. Any one type is present. And let us see the C, that is stomata on upper epidermis. Generally in majority of the monocots the stomata number will be less in the upper epidermis and more on the lower epidermis. Of course here in monocots on upper epidermis also we can see the presence of stomata. And D, that is stomata on both the epidermal layers. Here this type of nature, which here actually in both of them the stomata present on both the epidermis, but here in dicot leaf more number of stomata on the lower side, that is hypostomatous, and less on the upper side. Whereas in the monocot leaf almost the number of stomata will be equally distributed on both the upper epidermis and the lower epidermis. So here from the four options that have been given here, the differentiation of palisade and spongy parenchyma is present in dicots, but such type of character is not being seen in the monocotyledonous plants. So from the given options, option B is the correct answer. Question number 42. The grafting is not possible in monocots because the given options are: A. Have parallel venation, B. Lack cambium, C. Herbaceous, and D. Have a scattered vascular bundles. Generally in the monocots the plants does not exhibit secondary growth. And one more thing is that have to be considered here, it is not because of parallel venation, because the veins are present in the leaf, they are not concerned with the secondary growth or grafting process. And here one thing is, for grafting process it is a type of vegetative propagation where two different types of plants are grafted with each other. One is called as scion and other is called as a stalk. So the stalk and scion have to be binded with each other, and both of them are tied with the rubber for some days. After some days what happens, that the scion and stalk will get attached with each other. What is the reason why both of them are attached with each other means because of the activity of the meristematic tissue. So, which type of tissue are helping in that process means the cambium tissue, because cambium is a meristematic tissue which will start dividing and redividing to produce new tissue. The tissue of scion will be protruded into the stalk and the tissue of the stalk will be protruded into a scion. So, that is the reason why the grafting is possible. So, from the given options we can say that in monocots grafting is not possible because of lack of cambium. So, from the given options option B is the correct answer. Question number 43. Endodermis is a part of. The given options are A. Epidermis, B. Pericycle, C. Cortex and D. Medulla. So, here if you see the tissue systems present in the plant body, we can say that three tissue systems are there. One is called as epidermal tissue system, ground tissue system and vascular tissue system. In the ground tissue system, so we can see the presence of cortex there. And this cortex is generally divided into three parts. So, the outermost layer is called as hypodermis, second part is called as general cortex and third part is called as endodermis. This type of characterization can be seen in the dicot stem. And when we see the roots, either the monocot root or the dicot root, here in place of hypodermis we can see the presence of exodermis and the second part will be the same one, that is general cortex, and the third part will be endodermis. So, that is the reason why we can consider that. Okay, here one more thing, endodermis is being seen in the monocot and dicot plants, particularly in the dicot stem and roots of monocot and also in the dicot plants. And endodermis is considered to be the innermost part of the cortex. So, from the given options we can say that option C is the correct answer. Question number 44. The limiting layer of steel is. The given options are A. Epidermis, B. Endodermis, C. Piliferous and D. Epiblema. So, here if you observe the internal structure of any part, either the stem or the root, the outermost limiting layer is called as epidermis, outermost limiting layer of the plant part, and below this one we can see the presence of a cortex. So, here either if it is an stem, here we can see the hypodermis, and if it is a root it will have exodermis, and below this one we can see the presence of general cortex, and the innermost layer of cortex is called as endodermis. So, these three parts are combinedly called as cortex, and inside the endodermis whatever the tissue that is present we call it as a steel, and in the steel also the outermost layer of the steel which is present just below the endodermis, that part is called as pericycle, and of course inside this one we can see the presence of vascular tissue and central part is occupied by the medulla or pith. So, here as we are saying that it is a outermost layer of steel, but which part of the plant or which tissue of the plant are acting as a limiting layer means here the endodermis, which is innermost part of the cortex, is acting as a limiting layer for the steel. So, from the given options option B is the correct answer. Question number 45. Match the following. List 1. A. Bulliform cells, B. God cells, C. Lenticell, D. Subsidiary cell. List 2. A. Stomata, B. Aerating pore, C. Accessory cell and D. Isobilateral leaf. The correct match is option A. 1D, 2A, 3B, 4C. Option B 1A, 2D, 3B, 4C. Option C 1D, 2B, 3C and 4A. Option D 1A, 2B, 3C and 4D. So here let us take the list 1 and list 2. In the list 1, here starting with the first one, that is bulliform cells. Here bulliform cells are special type of cells which are being seen in the monocotyledonous plants, particularly in the grasses. So these are large sized cells which are helping in rolling and unrolling of the leaves. So here these are considered to be present in the isobilateral leaf. So why it is related with the isobilateral leaf means, as I said that bulliform cells are present in the monocotyledonous plants or leaves of monocotyledonous plants, and here the leaves or monocotyledonous plants are called as isobilateral leaves because the dorsal, there is no differentiation between the dorsal side of the leaf and the ventral side of the leaf. So here for A it is 4 is a correct match, and B, that is God cells. God cells are the modified epidermal cells which are surrounding a special aerating pore. That special aerating pore is called as a stomata, or we can say that they are helping in exchange of gases and also for a transpiration process. So these are the structures which are concerned with stomata, or we can say that these are the two specialized modified cells which are present around the stomata and helping in opening and closing of the stomata. Then the third one, that is lenticels. Lenticels are being seen in the woody matured stem, and these are helping in gaseous exchange, particularly in the matured parts of the plant body. So these are helping in aeration. So that's why they are considered to be the aerating pore. Then the last one, that is D, subsidiary cell. Subsidiary cell or subsidiary cells are also present around the God cells which are helping in opening and closing, where helping in turgidity and flaccidity of the God cells. So here these are considered with accessory cells for the stomata. So these are the accessory cells. So from the given options we can say that 1, A 4, B 1, C 2 and D 3 is the correct match. So from the given options, option A is the correct answer. Question number 46. Cambium activity is highest in the given options: A spring, B winter, C autumn and D rains. So here cambium is the one which is helping in secondary growth, and it may lead to the formation of annual rings also, or we can call this annual rings or the growth rings. And here here one more thing, that in different seasons the tissue formation, the amount of tissue that is formed will be different in the plants. So depending upon the need of the water and minerals, the activity of the cambium will be depending. Particularly if we take the spring season, where we consider that as a season where the new organs will be formed, that is leaf formation will be more. When the new organs or leaf formation is more, the requirement of water and minerals will be high. As the requirement of water and minerals will be high, there is a need of more number of xylem tissues and large sized xylem tissues. So that is the reason why at that time, that is in the spring season, the activity of the cambium should be highest. Because of that correlation or that reason, so we can see the such type of change during the spring season where the highest activity of the cambium can be seen. So because of the reason, so from the given options, option A is the correct answer. Question number 47. Generally, the secondary phloem remains functional for. The given options are A, less than 1 year, B, more than 1 year, C, for 1 year, and D, as long as the plant lives. So here secondary phloem which have been formed as a result of the secondary growth. It have been formed on outer side of the vascular cambium. So whatever the secondary phloem that is formed. Okay, one more thing that here every year the spring wood and autumn wood is formed, that is the growth ring will be taking place. It is, but in the phloem there is no such type of annual ring formation. Then also we can say that whatever the phloem, secondary phloem that have been formed by the vascular cambium will remain active for 1 year only. So here from the given options, option C is the correct answer. Question number 48. In a dicotyledonous stem, the sequence of tissues from outside to inside is. The given options: A, phloem, pericycle, endodermis, phloem. B, phloem, phloem, endodermis, pericycle. C, phloem, endodermis, pericycle and phloem. D, pericycle, phloem, endodermis and phloem. So in a dicotyledonous stem, the stem which undergoes secondary growth. So here let us see the tissues from outside to inside. Here they had given some tissue which are been considered in the question, but let us see what are the tissues which have been formed here. So the outermost part which is formed, that will be the phloem tissue. Below the phloem, the phloem is present, and below the phloem, phloderm. So these three are combinedly called as periderm tissue. And these are the tissues which are being formed as a result of extracellular changes taking place in the plant. But before that extracellular changes, some intracellular changes will also be taking place. And below the phloderm, we can see the presence of some primary phloem also. It may or may not be present because of the activity of, okay, before that we can say that here we can take the vascular cambium. The vascular cambium will produce some secondary tissue towards outside and some secondary tissues towards inner side. The one which is formed towards outer side is called as a secondary phloem, and the secondary phloem will be pushed towards upper side such that it will occupy the place of the primary phloem. And because of in some of the plants, because of the excess secondary phloem, the primary phloem may be completely crushed, or sometimes in some of the plants little amount of primary phloem will be left out. And below the vascular cambium, below this one we can see the presence of secondary xylem. And here this is about the cambium we are talking about, but before that at this region we can see the possibility of presence of pericycle, because pericycle is the outermost limiting layer for this one. So there is a possibility of presence of pericycle, and not only the pericycle, outside the pericycle we can see the presence of endodermis also. Outside the pericycle, endodermis is also been seen. So from the given options we can say that in the option C, if you see the phloem, this part is present, and next one will be the endodermis, below that one the pericycle, and below that the phloem will be present. So from the given options, the correct sequence is phloem, endodermis, pericycle and phloem. So from the given options, option C is the correct answer. Question number 49. Which of the following is related to plant structure is correct? The given options are: A. Cork lacks stomata, hence cannot carry out transpiration. B. Passage cells help in transfer of food from cortex to phloem. C. Young sieve tube elements possess cytoplasm. D. The shoot apical meristem has quiescent center. So here from the given options we have to identify the correct statement. So first one, let us see the option A. Cork lacks stomata. Actually cork is formed as a result of secondary growth, and the epidermal tissue which is present outside the cork will be ruptured. That's the reason why there is no possibility of presence of stomata. And when the stomata is absent, there is no possibility of transpiration. So no transpiration. So this statement is correct related to the question number 49. Let us see other options also, whether any possibility of other correct statements here. Passage cells help in transfer of food from cortex to phloem. Actually the passage cells are the cells of endodermis which lack Casparian thickenings or Casparian bands, and these are the one which will help in movement of water from cortex to stele, but not the food from cortex to phloem tissue. So this statement that have been given is wrong statement. And let us see the C statement. Young sieve cells. Sieve cells are the part of phloem tissue. Young sieve cells contains cytoplasm along with nucleus. So we can call this as a protoplasm. But one thing had to be considered here: when the sieve cells become matured, the nucleus will gradually disappears. At that condition we can say that only cytoplasm is present, but in the young cells protoplasm is present. So from the given options the statement is wrong. Let us see the statement D. The shoot apical meristem has quiescent center. Quiescent center is being seen in the root apical meristems. The root apex will show the presence of quiescent center. So from the given options this D is also the wrong statement. So from the given options statement A is the correct one. So option A is the correct answer. Question number 50. Bark refers to the given options: A. Phellem, phellogen and phellodum. B. Peridum and cortex. C. Phellem, phellodum and secondary phloem. And D. Peridum, cortex, pericycle and secondary phloem. So here bark is a tissue which is formed as a result of secondary growth in the dicotyledonous stems. Here one more point had to be noted here, that bark is nothing but all the tissue which is present outside the vascular cambium. Let us see what are the tissue which are present around outside the vascular cambium. Vascular cambium will produce secondary phloem towards outside, and there is a possibility of little amount of primary phloem also. It may or may not be present, but if it is present it have to be considered to be the part of phloem tissue. And outside that one there is a possibility of presence of pericycle, and outside the pericycle the endodermis is present, and little amount of the cortex tissue. We can say that some of the layers of inner cortex will also be remained there, and we know that cork cambium is nothing but the phellogen is formed from either the inner layers of cortex or middle layers of the cortex. At that condition, some of the cortex tissue will be left out here, and when cork cambium is becoming active, it produces secondary cortex or phylloderm towards inner side, and cork tissue towards outside, or we can call it as phylum. So these are the different types of tissues which are the part of the bark tissue, or we can say that from here onwards all the tissues, that is the tissues which are present outside the vascular cambium, are considered to be the part of the bark. So it contains cork, cork cambium, secondary cortex, some part of the cortex, endodermis, pericycle, little amount of phloem, and also the secondary phloem. So from the given options, the option D is showing the presence of periderm. Periderm is all these three. Then cortex, pericycle, of course endodermis is not given there, but they had given the pericycle, and secondary phloem is given. So from the given options, the four tissues that have been given are matching here. So from the given options, option D is the correct answer. Question number 51. Which type of vascular bundles are found in dicot stem? The given options are A. Collateral, conjoint and open with endark xylem. B. Radial with exarch xylem. C. Bicollateral, conjoint and closed. D. Radial with endark xylem. So here the general character of dicot stem. So let us see each vascular bundle in this one. If you consider this as a vascular bundle, so here on the lower side we can see the presence of xylem like this, and in the xylem also the metaxylem are towards the outer side, and protoxylem are towards inner side. So metaxylem and protoxylem. And our upper side, outer side of this vascular bundle, the presence of phloem tissue. And separating the xylem and phloem, there we can see the presence of cambium. This type of organization can be seen in the vascular bundle of dicot stems. So here both phloem and xylem are present in the same bundle, so we call it as a conjoint. And if we draw a line, both the xylem and phloem are coming on the same radii, so collateral. And separating the phloem and xylem, we can see the presence of cambium, so open type of vascular bundle. At the same time, the protoxylem is towards inner side and metaxylem is towards outer side. So this type of xylem arrangement is called as endark xylem. So the dicot stem shows collateral, conjoint and open with endark xylem. So from the given options, option A is the correct answer. Question number 52. Bulliform cells that help in rolling of lamina in drought are present in the epidermis of. The given options are A. Monocotyledonous grass, B. Dicot leaf, C. Both 1 and 2, and D. None. So here bulliform cells, otherwise called as motor cells. This type of characteristic feature can be seen in the upper epidermis, where some of the cells will become larger in size. This type of special cells which are present in the upper epidermis are called as a bulliform cells, and these are the one which will help in rolling and unrolling of the leaf depending upon the availability of moisture and dry condition in the atmosphere. This type of characteristic feature is being seen in the monocots, particularly in the grasses. So from the given options, option A is the correct answer.